Hi, and welcome to Microbiology. My name is Dr. Sheila Taylor, and I'm going to be your facilitator for this semester's material. Before we start Chapter 1, just some housekeeping items. First of all, every chapter of your text begins with what's called microbes in the media. And these are linked to articles that are found in media, and I will ask you to read some of these as we go through, and I will give you instructions on how to access these in the classroom. Secondly, I want to give credit to McGraw-Hill, your publisher, for many of the pictures, figures, and charts that you will see in these PowerPoints. So, let's get started. And let me get my laser pointer on here so that we're together. The first, microbes in the media, is talking about coffee makers, specifically Keurig coffee makers, but I suspect that many coffee makers are probably have the same issues that the Keurigs in the articles actually have. And this is the fact that they have many microbes that are actually in the coffee makers themselves. This means that you're probably taking in some of these with your morning Java. So, in looking at the scope of microbiology, really in this first chapter, we're going to talk about the things that we're going to talk about for the rest of the semester. In public speaking, one thing that speakers are taught is tell your audience what you're going to tell them, and then tell them what you told them. So this is really the introduction. So the first question is, what is microbiology? Microbiology is the specialized area of biology that deals with organisms that are too small to be seen with the naked eye. In other words, we must actually look at them under the microscope. These can be called microorganisms or microbes, which is the primary thing that I will use this semester, but they can be known as germs, viruses, or agents or disease-causing agents, or another term is pathogen, or simply bugs. The major groups of microorganisms that we are going to cover this semester include the bacteria, protozoans, fungi, the viruses, which we've heard a lot about viruses in the past few months, prion diseases, which you may not be as familiar with, the helminths, which includes the worms and the flukes, and even though in the adult stages you can see these worms or flukes oftentimes, part of their life cycle is going to be microscopic, and that's why we include them in the study of microbiology. And finally, the algae. We're not going to spend a lot of time with the algae simply because most algae do not cause major disease processes, but they are included in the microbiology field of study. Microbes are easy and difficult to study. And part of this is because they are so small that we have to use the microscope, and yet they're everywhere, and so we can actually find them pretty much anywhere that we look. They typically reproduce very rapidly. They typically have very large populations, and most of them can actually be grown in the laboratory in some way or some form. They can't be seen directly with the naked eye, therefore they must be analyzed through indirect methods in addition to using microscopes. Microbiologists actually study a variety of different things in the microbes. One, we can look at their cell structure and function. We can look at how they grow and the physiology in which they grow with, their genetics, the taxonomy and their evolutionary history, and how they interact with living and the non-living environments. Microbiology includes various branches of study: medical microbiology, public health microbiology and epidemiology, immunology, industrial microbiology, and agricultural microbiology, along with environmental microbes. This table in your text actually goes through some of the different areas, such as with medical microbiology. This deals with microbes that cause disease in humans and animals. Researchers examine factors that make these microbes that cause disease or how they cause disease and the mechanisms for inhibiting them. Public health microbiology and epidemiology we have heard a lot about in the recent months with COVID-19. Institutions involved in this include the U.S. Public Health Service, Centers for Disease Control and Prevention, the World Health Organizations, localized health groups, especially like Greene County Health, and other groups as well. Immunology. This really studies how the body responds to disease when it occurs. And we'll spend a couple of chapters looking at immunology in the body. Industrial microbiology, we're not going to spend a lot of time with, but certainly this branch safeguards our food and water. It also includes areas of biotechnology. And so we look at gene therapy and genetically modified organisms in the industrial microbiology area. Agricultural microbiology includes looking at relationships between microbes and the domesticated plants and animals. And environmental micro looks at how microbes interact with the environment. And this can include both water, soil, and even looking at the generalized crust of the earth in all of these areas. In these PowerPoints, you'll find concept checks, and these are actually questions that I look at when I sit down to write an exam. It doesn't mean they'll all be on the exam, but these are good study questions. And so I'm just going to post them here, and I might mention a few of those as I go through. So certainly you should know what types of microorganisms we're going to look at in this semester. What types of cells that viruses can infect, and we'll spend a whole chapter on viruses, and then looking at those branches of microbiology. Now, what is the importance of microbes on Earth? Well, the microbes have actually shaped the development of Earth's inhabitants and the evolution of life forms for billions of years. Single-celled organisms arose approximately 3.5 billion years ago and were the only living inhabitants until about 2.9 billion years ago. The bacteria, the archaea, and the eukaryotes arose from this last universal common ancestor. This was the precursor cell for all of the cell life that we see today on Earth. Eukarya, the EU, is actually Greek meaning true or good, and karya refers to nucleus. So eukaryotes have a true nucleus. This means that their genetic material is surrounded by a nuclear membrane, and they also contain membrane-bound organelles such as the Golgi apparatus, endoplasmic reticulum, mitochondria. Pro means before or pre, and so a prokarya has a pre-nucleus, or in other words, their genetic material does not have a nuclear membrane, nor do they have membrane-bound organelles, and these have traditionally been classified as prokaryotes. However, there are distinct differences between the two major groups of prokaryotes, the bacteria and the archaea. And some scientists are actually now calling these types of cells akaryotes because they lack a true nucleus. This figure just shows the probable origin of evolutionary life on Earth. Prokaryotic cells are ubiquitous. That means they are everywhere. They have been found deep in the Earth's crust. They've been found deep in the polar ice caps. They're found in all layers of the ocean, from the surface to the deepest parts, and certainly inside the bodies of plants and animals. So we can actually say that prokaryotes, the microbes, are found pretty much anywhere you want to look. Microbes have been deeply involved in the flow of energy and food through the Earth's ecosystems. Bacteria, some of them are what we call anoxygenic photosynthesizers, meaning that they use light to produce their energy, but they do not produce oxygen in the atmosphere. The production of oxygen, though, by microbes that do that process have allowed for species diversification. Photosynthetic microbes account for more than 70% of Earth's photosynthesis, contributing therefore to the major amount of oxygen that we see in our atmosphere. Algae being one of the largest groups of these microbes that contribute to the overall atmospheric oxygen content. And so I put in a nice little picture of good old pond scum. And though it may look kind of disgusting, it's actually very important in the ecosystem to keep the oxygen levels in the atmosphere. Another thing that's very important with many microbes is decomposition. Without decomposers, we would literally be neck deep in organic waste material. And so these microbes break down dead matter and wastes and recycle the energy and the nutrients found in the dead organic material back into the atmosphere. And this is primarily accomplished by the decomposer bacteria and fungus. Microbes are the main force that drive the structure and content of the soil, the water, and even our atmosphere. Certain gases are produced by the microbes, such as, again, oxygen by the photosynthesizers. Microbes that live within the Earth's crust actually shape the crust of the Earth. Bacteria and fungi living in complex associations with other plants and animals actually help feed the population of the world. So, you should definitely know the difference between a prokaryote and a eukaryote. And here's a good true and false question. Microscopic organisms produce more oxygens than plants. Another one, more than 50% of the microbes on the planet live below the Earth's crusts. How have we used microorganisms? Well, we've been using microbes for thousands of years to improve life and even shape civilizations. Think about it. Without yeast, what would we be? Production of beer and bread, the staples of life. That's what we use yeast for primarily. Other fungi have been used for centuries for the production of cheese, and moldy bread was even used in Egypt to treat wounds. And we'll talk about why that was probably actually very effective later on. Today, we can manipulate the genetics of these microorganisms and actually use them in industrial or other settings. And in genetic engineering, we can actually manipulate the genetics of microbes, plants, and animals for the purpose of creating new products or genetically modified organisms. And I'm here to tell you, without genetically modified organisms, we would not be feeding the world's population today. We'll talk a little bit about recombinant DNA technology. These techniques allow the transfer of genetic material from one organism to another and deliberately alter the DNA of the recipient organism. And bioremediation. This is the introduction of microbes into the environment to restore stability or to clean up toxic pollutants such as oil. There is a group of oil-eating bacteria that actually will munch up the oil and get rid of it. When the Deep Horizon exploded a few years ago in the Gulf of Mexico, the environmental impact would have been so much greater if we have not had these oil-eating microbes, which they took out and sprayed on the oil slicks before they reached our shores. So, you should be able to name 3 products produced by genetically modified organisms that benefit humans. We'll talk about those a little bit later on as well. You need to describe one method in which microbes are used for bioremediation. Now, as folks in the human healthcare system or in medicine in general, we get so focused in on a very, 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 very small group of microbes that are known as the pathogens. A pathogen is any agent such as a virus, bacteria, fungus, protozoan, helminth that causes disease. The vast majority of microbes are non-pathogenic. Only a very, very small percentage of our microbes found in our world are actually pathogenic. Nearly 2,000 different microbes have been identified to date that can cause disease in either animals or humans, and we do share a lot of diseases in common with our animals. Of those 2,000 different microbes, we share in common almost 1,600 of those that cause the very same or similar diseases in our animals. This chart just compares the death rate in the United States to the death rate worldwide. And you can see that heart disease is number one. And this was from the year 2015. But heart disease actually still remains number one around the world. Cancer is the second leading cause of death in the United States, and stroke worldwide is the second leading cause. But what I want you to notice about this is those things in red: influenza, pneumonia, diarrheal diseases, tuberculosis. All of these are actually caused by pathogens. And so, again, we get so focused in sometimes on the few microbes that cause pathogens because here they are on the top 10 causes of death. Communicable versus non-communicable diseases, or infectious versus non-infectious diseases. Communicable diseases simply are those diseases that are easily transmitted from one individual to the next susceptible individual. Non-communicable diseases are not transmitted in this way. This was actually a study done several years ago, but it still holds fairly true, and they looked at communicable diseases. And this included also maternal and perinatal conditions and nutritional deficiencies that are in the blue. And then they looked at chronic diseases, which include cardiovascular disease, cancers, chronic respiratory disorders, diabetes, etc., of which you can see that many of those are in that top 10 list of causes of death. And then they just looked at accidental injuries in the green. Then they looked at the death rate from each of these categories in low-income countries all the way up to high-income countries. Now, communicable diseases, you'll notice that in low-income countries, we have a lot of deaths from those communicable diseases or maternal perinatal conditions or nutritional deficiencies. Why might that be? We'll actually discuss this in class. The chronic diseases, well, deaths from those do tend to be higher in those lower income countries, less so in the higher income countries. What I found interesting, however, was that the upper middle income countries actually have the lowest death rate from chronic diseases. And then injuries kind of follows again with the communicable diseases in income. For example, malaria. Malaria is a protozoan parasite that kills between 700,000 to 1.2 million people every year. Estimated that basically one person every minute in the world dies from this protozoal diseases. It's transmitted by the Anopheles mosquitoes, and you'll see that these kind of ring-like structures in these red blood cells, these are actually the malaria protozoan parasite, and so it destroys the red blood cells and causes severe anemia. Prevention of infection of malaria is through the use of bed nets, although quite inexpensive. They are actually typically they are much too expensive for very poor families in regions where the vector, the Anopheles mosquito, is found. And that's primarily in the tropical regions of Africa, India, and parts of actually South America. We also have to look at new or emerging diseases as well as older diseases that are now increasing. And so we have things like Ebola, West Nile virus, Zika virus. We can look at AIDS, hepatitis C, viral encephalitis, such as St. Louis encephalitis or West Nile virus. Polio, leprosy, parasitic worm diseases have primarily been eradicated, but we still see them in some areas of the world. Certain diseases once considered non-infectious are now found to be caused by some microbes. So for example, gastric ulcerations, 90% of all gastric ulcerations will have a concurrent Helicobacter pylori bacterial infection. There are links between certain cancers and certain bacteria and viruses. The Coxsackie virus that causes hand, foot, and mouth disease has now been associated with the onset of diabetes. The Borna agent has actually been linked to certain mental dysfunctions. And multiple sclerosis, OCD, coronary artery disease, and obesity have been linked to chronic microbial infections. Interestingly enough, there have been several studies that said that obese patients typically have a different normal gut microbiome than thin people. And I wish they would figure out how they could get that skinny microbiome into me. Today, the subtler side of microbes and the quiet, slow, and destructive disease that they cause are still being discovered. So for example, female infertility oftentimes is caused by chronic sexually transmitted infections, and the number one culprit of this is chlamydia. Liver cancers linked to several of the hepatitis viruses, and cervical cancers linked to the human papillomavirus. We now believe that many cancers may actually have a viral linkage. An increasing number of patients with weakened immune systems are subject to infections by common microbes not pathologic to healthy people. You actually do inherit your immune system, or you can have a disease that will weaken your immune system that will actually make you more likely to become infected with another disease. And of course, we're going to talk about drug-resistant microbes, which also contribute to the increase in infectious disease. So, true or false? All microbes cause disease. Why is there a difference between the top 10 causes of death in the U.S. versus worldwide? And name 3 connections between diseases considered to be non-infectious and a microbe. So let's look at some general characteristics of the microbes. And we'll look first at the archaea, the bacterial and archaeal cells. Bacteria and archaea tend to be approximately 10 times smaller overall than eukaryotic cells. They again lack the membrane-bound organelles such as mitochondria, endoplasmic reticulum, Golgi apparatus, and they do not have that membrane-bound nucleus. The majority of all microorganisms are single-celled, and this includes all of the bacteria, all of the archaea, and many actually of the eukaryotes. So here again are some of the pictures of our microorganisms that we're going to actually look at, including the bacteria, the viruses, protozoans, helminths, fungi. This just simply compares the relative size and some of the major structures of the prokaryote and the eukaryotic cells. Bacteria and archaea, again, they do have a chromosome. Typically, it is a single circular chromosome that is haploid in number. They do have ribosomes. Ribosomes, if you remember, are organelles, but they are not membrane-bound organelles. Can anybody remember what the ribosomes do for the cells? Whereas eukaryotic cells have the true nuclear membrane, they have the membrane-bound organelles, along with ribosomes as well. And then we'll look at something that I refer to as an acellular structure, and that is the viruses. Viruses are simply made up of a nucleic acid, either DNA or RNA, never both, surrounded by a protein capsule known as the capsid, and may have a phospholipid envelope or not. Helminths, again, are typically not microorganisms in their adult stage, and this is actually a scarce lumbricoides, the common roundworm. But They are included in the study of infectious diseases because they are transmitted similarly to bacterial diseases, and parts of their life cycle are typically microscopic. This is actually the egg of the roundworm, and you would only see that under the microscope. The human body will respond to these helminths in the same way as it would respond to a bacterial disease to try to get rid of the foreign entity. So, the majority of microbes live in habitats such as the soil and the water. The vast majority of microorganisms are harmless, do not cause disease, and oftentimes are actually very beneficial for our use. Parasites are those small number of microbes that can be harbored and nourished by the host. The problem is that while they receive their nutrition from the host, they cause damage and disease in the host. So, in terms of size, how are eukaryotes different from the bacteria and the archaea? Give 3 examples of the eukaryotic organelles, and there's more than 3, and be able to explain why helminths or the worms and flukes are considered in the study of microbiology. Now, early ideas about disease transmission were very difficult because people couldn't see the microbes. They understood that certain foods would spoil, they would become inedible, or they would cause illness and disease. For example, think about the Black Plague in the Middle Ages, or smallpox that has been now eradicated pretty much from the world. But they understood that these diseases were caused by some kind of transmissible matter. What they didn't understand was that these were caused by microbes that were either transmitted from one person to another in the case of smallpox, or through some sort of vector, in this case the rat flea with the plague. And so many of the early individuals in studying disease came to believe in a theory known as spontaneous generation. And what spontaneous generation truly said was that living matter could come from non-living stuff. So, early on, Robert Hooke in the 1600s actually made the first observation of microbes. He was looking under a magnifying glass at plants, and what he described were little boxes. These were actually the cells of the plant. Anton von Leeuwenhoek came along just a little bit later, and while others had made, quote, crude microscopes, what Leeuwenhoek did was made a crude microscope that examined threads in fabrics. He also then looked at other things, and every time he looked at a new thing, he made a new microscope. And he made drawings of what he called animalcules in rainwater and what he'd scraped from his teeth, and we now know that was plaque today. Anton von Leeuwenhoek is considered the father of modern microscopy, simply because even though his microscope had a single lens, he was the first that had a stage that was able to focus up and down on a particular entity. What Robert Hooke saw were the plant cells, but he could only have them in focus at one focal point. He could not go up and down through all of the layers of the cells. And so this is what really made Leeuwenhoek the father of modern microscopy, was the fact that he could actually now focus up and down using his very crude microscope. And this is an example of what his microscope looked like. Actually, what he did was use a pen. And so he would put his specimens actually on the tip of the pen, but the focus screw allowed him to move up and down. And so he could look at different layers of what was on the tip of the pen. And this was actually what then he drew. And if you look at these, they actually look a lot like bacterial cells that we see today. Modern microscopes, such as what you use in the laboratories, are much more complex. They typically have more refined lenses, and lenses is plural. A condenser, which will focus light onto the stage and the specimen you're looking at, finer focusing devices, and typically now built-in light sources. Student microscopes, however, are not greatly different in structure and function than even Leeuwenhoek's microscope. It simply is now that we have a finer detail that we can see. I borrowed this from a text that just basically put some years on some of the major discoveries in microbiology. And really, microbiology is a young science, with Robert Hooke in 1665 making the observation of the plant cells, Leeuwenhoek a few years later with his microscope. Almost 50 years some later, in 1735, Linnaeus was the first one that began taxonomy. And basically, he put all living organisms into two main categories, the kingdom Animalia and the kingdoms Plantae. 1798, Edward Jenner was the first one to do a true vaccine, and we'll talk about him later on. Schimmelweiss, I'll talk about in this chapter. He was actually came up in 1840 with the dramatic idea of washing hands between patients. It was such a radical idea, he was almost drummed out of the Austrian Medical Society because how dare he call doctors dirty. But what he found out was that women that died of childbirth fever after having children, actually the death rate dropped when the physicians simply washed their hands from going from patient to patient. Pasteur. Louis Pasteur is a big name in the golden age of microbiology in the mid-1800s. First of all, he was the one that completely disproved spontaneous generation. We'll talk a little bit about that. He was the first to really understand fermentation. And of course, we've honored him with giving the name of heating juices and milk today destroy microbes with the process of pasteurization. Lister was the first one who actually developed an aseptic for using in surgery, and we've given him the honor because basically the product you buy today, known as Listerine, is based upon the aseptic that he used in surgery. Koch, also a big name. He was the one who actually came up with the germ theory of disease, linking a specific microbe to a specific disease. And continuing on into the 20th century, Watson and Crick saying in 1953, this is what we think that DNA looks like at a molecular level. And then Woosin set 1978 was the one who actually came up and divided the prokaryotes into bacteria and the archaea and came up with the three-domain system that we still use today. So, what was this spontaneous generation? Well, think about it. So, you moved into a new hovel, and over there in the corner, there was nothing. But you started to throw all your trash and everything in that corner, and all of a sudden one day there were mice. Well, where did the mice come from? Well, the spontaneous generation individuals said that trash wasn't there, no mice. The trash is there, now there's mice. So the mice had to come from trash. Again, they didn't really understand the whole process of a living organism. Spontaneous generation is also known as abiogenesis, a without, bio, life, generation of. However, what we find is that most spontaneous generation individuals said that living organisms arose from the non-living matter and that a vital force had to endue that non-living matter with a life force. The competing theory was biogenesis, and the debate between these two raged for some 200 years. Biogenesis stated that only living organisms can arise from pre-existing organisms. So, 1745, John Needham put boiled nutrient broth into covered flasks. What he did is he actually took a meat broth and he boiled it. Even in the 1740s, they understood if you boiled it, you got rid of stuff. Okay? We still didn't know what microbes were, but if you boiled it, it got rid of disease-causing stuff. He then poured it into flasks and sealed the flasks, and this was his result. So he boiled the broth, then placed it in the flasks and sealed this, and what he found in a few days was that there was growth. Now again, he didn't know it was microbial growth at the time, but he could see stuff was growing in there because of the turbidity. And so John Needham said, well, there wasn't anything living in there because I boiled it. So he came down on the side of spontaneous generation. Well, this guy came along a few years later, and for the life of me, I can't pronounce his name, so I just call him Dr. Larry. But he actually did the same experiment that Needham had done. He boiled the nutrient broth, but what he did was he put the broth in the flasks, then boiled it, then sealed them. And he saw no microbial growth. Well, what the spontaneous generation people said about this gentleman's experiment was, well, the flasks were sealed, therefore that vital force couldn't get in and endow that boiled broth with the life force that needed to grow living organisms. And he goes, but Needham sealed his flask too, and it still did it. Well, the life force got in before he sealed it. Okay. In 1861, Louis Pasteur came along, and he was the last one that really put the last nail in the coffin of spontaneous generation. He demonstrated that microorganisms were present in the air. And he did this again, really building on Needham and Dr. Larry's experiment. He took nutrient broth, placed it in a flask, heated it, but did not seal it. He took another one, did the same thing, and then sealed it. In the flask that was not sealed, there was microbial growth that was seen. In the ones that were sealed, no microbial growth. But again, the spontaneous generation people said, well, you sealed it. It's like vital force couldn't get in. Vital force could get into the one that was not sealed. So Pasteur went a little bit further. And he developed a S-shaped flask. And this is what they looked like. It had a long, thin neck on it. He poured the broth in, boiled it, heated the glass of the neck so that it formed this S-shaped curve. And then he let it sit for months. And no microbial growth was seen. This was open. It was not sealed. And so what he saw was the fact that, life force could get in, but it couldn't actually make that curve. And so the microbes just simply would never get down into the growth media. Whereas if he took the boiled media, broke the neck of the flask off, then microbes from the air could actually get in and microbes could occur. The spontaneous generation people just could not refute this because this was open, couldn't get in, so no growth occurred. So Pasteur was really the last one that said that spontaneous generation was incorrect and the theory of biogenesis was the correct theory. So Pasteur made many contributions to microbiology. We'll talk about Robert Koch here in just a few moments. And I'll even mention Rebecca C. Lansfield, who only passed away in 1981. She was the first to classify Streptococcus according to serotypes. And this is actually strains or variants within the same species of organisms. In 1970s, we discovered restriction enzymes that will cut DNA at certain spots. In the 1980s, the invention of the PCR technique happened. And this is just simply a process of copying DNA and making millions of copies from a single copy in a very short period of time. And we'll talk about PCR In the 1980s and beyond, the importance of biofilms were seen in infectious diseases. And in the 2000s, and in fact in the year 2000s, we mapped the human genome. And we've also discovered the importance of the small RNAs, especially in eukaryotic cells. So, from the mid-1600s to the present, science is an ever-evolving collection of new information, and microbiology really is a very young science in the biology. So, let's talk a little bit about scientific method. This is the general approach taken by scientists to explain a natural phenomenon. In scientific method, you have to come up with a hypothesis, and this is a tentative explanation to account for what has been observed or measured. Now, deductive reasoning uses the general principle to explain specific observations, while inductive reasoning is the process of discovering general principles by careful examinations of specific cases. So, for example, the people that observed that mice came from the trash heap were using deductive reasoning. The trash heap wasn't there, no mice. The trash heap was, there mice. Inductive reasoning, think of Pasteur. He actually looked at looked at general principles, applying them to very specific incidences. And so he made his observation through experimentation. And inductive reasoning is a discovery process then that leads to the creation of a general principle. So here you go. Deductive reasoning. Knowing that opening attachments from unknown senders can introduce viruses or other bad things to your computer, you choose the specific action of not opening the attachment. Inductive reasoning, though, you have performed the specific action of clicking on an unknown attachment 3 different times, and each time your computer crashed. This leads you to the conclusion opening unknown attachments can be damaging to your computer. The scientific method can be a very lengthy process of experimentation, analysis, and testing. It eventually leads to conclusions that either support or refute the hypothesis. If experiments do not uphold the hypothesis or some part of it may have to be completely rejected or modified. So it's either discarded or modified to fit the results of the experiment. If experimentation supports the hypothesis, it's not immediately accepted as fact. The hypothesis must be tested and retested with consistent results. The results of these experiments then must be published and repeated by other investigators. A theory is a collection of statements, propositions, or concepts that explain or account for a natural event. The entire body of ideas that expresses or explains many aspects of the phenomenon. But a theory is not a law. A viable declaration that has stood the test of time and has yet to be disproved by serious scientific endeavors can remain a theory. The law of gravity is not a theory. You jump off a cliff, you're going to fall. It happens every time, no matter what. Okay? However, other things out there are theories, simply because they've not been disproved, but they can't be completely proved either. Now, science and hypotheses and theories must progress with technology. Advances in instrumentation allow new, more detailed views of living phenomena. We went from the light microscope to the electron microscope. We can now visualize under the microscope the atomic level. So we've gone from Hooke's little boxes in the 1600s to now being able actually to see an atom. Old theories must be re-examined and altered, and new ones proposed with new technology. So, what are the characteristics of effective scientists? And in medicine, that's what we are, is scientists. First of all, we have to have a curiosity. As a clinician, I'm curious as to what's causing the disease in my patient. I have to be open-minded. I can't just say, well, it's this and that's all that it can be. Have to think outside the box sometimes. We have to have skepticism. We can't just say, oh, he's an expert, so therefore he must always be right. Or, well, she's done this, so therefore she must be right. We actually have to have a good, healthy dose of skepticism. Creativity. We need to be able to come up with these hypotheses and experimentations to actually test them. And we need cooperation of others in our scientific field and even outside our scientific field. A readiness to revise their views of natural processes as new discoveries are made is quite necessary to be an efficient scientist. So here's the scientific method. And again, it doesn't always necessarily go in this order, but oftentimes the scientific method starts by asking a question. Where did the mouse come from? Okay. We can do background research. Well, we know that mice actually have mice. But where did the mice come from in my hovel? So can I construct a hypothesis? So I didn't have trash, no mice. I now have trash. The trash must give rise to the mouse. But I need to then test that hypothesis with an experimentation. Okay. If the experiment worked, okay, then I can analyze the data and draw conclusions. And if the results align with my hypothesis, I can communicate results to others where they then will actually do experimental data, oftentimes the same experiment, to see if they come up with the same information. Or did the procedure not work? Then I need to go back and troubleshoot my procedure, carefully check that all steps and setups are correct. And then what if my data does not align with my hypothesis? Well, then I either need to completely reject the hypothesis or modify the hypothesis, and then I can start over. And again, it always comes down to communicating results. But as I said, not everything has to be in this order. Sometimes we come up with an experiment, and then that experiment leads to the question, which then constructs the hypothesis. So, not a specific order, just simply a flow chart of a way that we can do the scientific method. Now, early experiments showed that microbes again are everywhere, and this is actually the end of a pen and a straight pen. And so you can see microbes are all over that tiny, tiny little tip. Air and dust are full of microbes. The entire surface of the earth, its waters, and all objects are inhabited by them. Even our own bodies are inhabited by microbes. John Tyndall found that microbes in the dust and air have a very high heat resistance. Ferdinand Kohn discovered and described bacterial endospores, which are a survival mechanism that some microbes can actually make. And the discovery of these endospores played an important role in the development of techniques of sterilization and also in the rejection of spontaneous generation. Robert Koch, I said we'd mention him again. He was the first to link a specific microorganism with a specific disease. Dr. Oliver Wendell Holmes actually observed that mothers who gave birth at home experienced fewer postpartum infections than mothers that went to the hospital to give birth. And that's where Dr. Ignaz Semmelweis came in. He showed that women became infected in the maternity ward in the hospital after examinations by physicians who had been working in the autopsy rooms came in to examine them without washing their hands. And so we can thank Dr. Semmelweis for the radical idea of washing our hands between patients. Mentioned Joseph Lister already. He was the first to utilize hand washing and the misting of operating rooms with antiseptic chemicals. His techniques became the foundation for the modern microbial control that we still use today. Pasteur can't not say enough about Pasteur. Invented pasteurization, showed that human diseases could arise from infection. Robert Koch establishing that series of proofs that verified the germ theory of disease. And he actually came up with 4 what's now known as Koch's postulates. He suspected the causative agent must be found in every case of a disease and be absent from healthy hosts. The agent had to be isolated and grown outside the host. When the agent is then introduced from those cultures into a healthy susceptible host, the host must get the same disease, and then the same agent must be found in the diseased experimental host. And so what we utilize is we associate the microbe with the disease, we isolate it from a sick individual, we then we isolate and grow it, and then inoculate it into an experimental organism, and then re-isolate and compare to the original organism we isolated. If they are the same, we can then state that organism caused X disease. So here we have a mouse with a disease, and we isolate the organism from the disease. We inoculate and grow a pure culture of it, and we take one of those colonies that's a pure culture and inject it into a healthy host. And the host then gets the disease or dies, and we re-isolate and compare again the 2 isolates, and they're exactly the same. So we can say this organism caused this disease. Now, Koch's postulates are still used today to prove the cause of infectious disease. However, there are some limitations to Koch's postulates. One, some microbes have very unique culture requirements and therefore have never been grown outside of a living host. They cannot be cultured on an artificial media, therefore we cannot isolate a pure culture. Some pathogens can cause several disease conditions, and in fact, the disease that Koch worked with, cutaneous anthrax, Bacillus anthracis, the causative agent, actually can also cause an inhalation form and a GI form of anthrax. And some pathogens cause disease only in humans, leading to some ethical issues for the postulate number 3. If this disease only occurs in human, do you want to be the experimental human, especially if this is a lethal disease? So while we still use Koch's postulates, we have to make some considerations for these 3 areas. So you should know who disproved spontaneous generation for the final time. Polymerase chain reaction, or PCR, when was it first discovered? When a hypothesis is supported by a growing body of data and survives rigorous scrutiny, then it's called a what? And aseptic techniques were actually first developed by whom? Now, we also want to identify these organisms, and with that, we want to put them into categories. And so, nomenclature. We use an array of new, unusual, and sometimes confusing names for organisms. Some organisms are named for their discoverer, some are named for where they're found, some are named for their shape, some are named for the arrangement of the organism, some are named for where they were originally found in the world. So there's a whole bunch of different ways that we name these organisms. However, the names are Latinized and therefore are used worldwide. So if I were to say someone who spoke only German, Escherichia coli, they would still understand Escherichia coli. Escherichia coli is named for its discoverer, Dr. Theodor Escherich, and its habitat, which is the human colon. They're Latinized because Latin is a dead language. It is not changing, and therefore anyone would recognize these Latinized names. The understanding and appreciation of microorganisms will be improved by learning a few general rules about how they are named. So, what is taxonomy? It is the science of classifying living organisms. It was actually first developed by Carl van Linnaeus in the 1700s, but has been certainly improved and changed over the intervening years. It lays down the basic rules of classification. In Linnaeus's times, all living organisms were either plants or animals because, again, they didn't really have the microscope truly yet. And so what taxonomy does is it establishes taxonomic categories of how things are related to each other. And it's useful in categorizing the 2 to 10 million or more different kinds of microorganisms that we see on the planet. So classification attempts the orderly arrangement of microorganisms into taxa or groups in how are they related to each other. The process of discovering and recording the traits of an organism so they can be recognized and placed into a taxonomic scheme. And so, for example, if I have a patient that comes in with a disease, I want to identify the causative agent of that disease so that I can then look at how can I treat that disease. So by identifying it into a particular group, then I know that I can use whatever treatment that it would be working for this group. We typically, for most organisms other than the viruses, use what we call a binomial system, bi meaning two, so two names. It's a combination of a genus name and a species name. The genus name is always capitalized, the first letter, and the species name always begins with a lowercase letter. Both names should either be italicized when in print or underlined when written by hand, but never both. So, for example, Staphylococcus aureus. Staphylococcus is the genus name, therefore the first letter is capitalized. Aureus is the species name, first letter is lowercase. So you can either allow it to be italicized, or if you're writing it, simply underline it. Now, we can sometimes use abbreviations because how many of you are going to learn to spell Escherichia? I still have to sometimes look it up. So the genus name can sometimes be abbreviated to save space, or if the genus name has been previously stated or referenced. Most of the time, E. coli is enough. We know it's Escherichia coli. We don't have to have stated it somewhere previously. But in literature, oftentimes what you'll see is the first time we name an organism, such as Staphylococcus aureus, after that, it can simply be abbreviated as S. aureus. Levels of classification, again, have changed over the millennia. We now work under the 3-domain system, and we'll talk about the domains here in a moment. The kingdoms are underneath the domains, and actually right now, the only domain that does have kingdoms is Eukarya domain. Then under each kingdom, there's a phylum or division. Under each phylum, there's a class. Under each class, orders. Under each order, family. And then comes the genus, the species, and even now, strains of each species. We're going to primarily focus on the domains. I'm going to mention the kingdoms. I'm not really going to go much into all of the different ones until we get down here to the genus, species, and a few of the strains. So when we look at classifications, here's the domains. And our 3 domains right now are bacteria, archaea, and eukarya. The bacteria and archaea are prokaryotic cells. Eukarya are, needless to say, eukaryotic cells. Underneath eukarya, we have 4 kingdoms: Animalia, Plantae, Fungi, and the one not listed here is Protozoa. So if I've identified something as a eukaryotic organism, and it's not a plant, it's not a fungi, it's not a protozoan, it's an animal, then I can look under the phylums: Chordata, Arthropoda, Platyhelminthes, Nematoda. And each one of these has specific categories. So Chordata means it has a backbone. Arthropoda, joint-legged animals. Platyhelminthes, it's segmented worms, and the Nematoda are unsegmented worms. Well, I've identified this organism as a eukaryote. It's in the kingdom Animalia, but it has jointed legs. So then I look at the classes under the jointed-legged animals in Arthropoda, and we have Insecta, Crustacea, and Arachnida. This organism has 8 legs. Insecta class has 6. The Crustacea has sometimes no legs to many leg-like structures. The arachnids have 8. Under the Arachnida, then we have several orders. This one happens to fit into the Parasitiformes, which include the mites and the ticks, or at least some of the mites and the ticks. And in the family, Exodiae, which are the hard ticks. And then I can look at the genus, and this is not all the genus of the hard ticks, but I know that this particular tick I'm seeing is Exodes. And then I can look at the species, and this happens to be Exodes scapularis, which is the vector for Lyme disease. Phylogeny is actually the taxonomic scheme that represents the natural relatedness between groups of living things. And we can actually form something that we call a cladogram. These are based on evolution, and so we go back to the earliest cells and then simply work our way up the tree. And so how is everything related? So in the animals, in the Animalia kingdom, we have a whole bunch of different things because they're all animals. They're not plants, they're not fungi, they're not protozoa. And so they're in that particular kingdom because they are all related to each other. Charles Darwin and Ernst Haeckel actually continued Linnaeus's thought process of the two kingdoms of plants and animals, and this was based on the morphologic characteristics of both. The third and fourth kingdoms, named by Haeckel, were actually added in the 1870s, and this included Protista, which basically was under Eukarya, which would contain anything that wasn't a plant or an animal, and Monera were all of the microbes. Robert Whittaker added a fifth kingdom a little later on, and this was fungi under the domain of Eukarya. And so now we had the plants, the animals, the Protista, which was kind of the catch-all drawer, and the fungi. All kingdoms encompass the two cell lines of prokaryotes and eukaryotes. Whittaker's system became standard in the 1960s, but Carl Woese and George Fox came along in 1978 and studied the small subunit of the ribosomes in the archaea and the bacteria and realized that there were differences. And so they proposed a separate taxonomic unit known as the archaea. And this was the first time then that we saw the three-domain system, with bacteria and archaea being the prokaryotic cells, but now being different classifications, and all of the eukarya. And so until 1978, we had the 5 kingdoms of Animalia, Plantae, Fungi, Protista, or the protozoans, and Monera, which were all of the microorganisms of bacteria and archaea. It wasn't then until 1978 that the 4 kingdoms of Plantae, Animalia, fungi, and Protista, or again now called protozoa, were listed under the Eukarya domain, and bacteria only contained 1 kingdom, and that was Monera. But since there is only 1 kingdom, we've pretty much gotten rid of the domain Monera, or sorry, of the kingdom Monera in bacteria. So this just simply shows the cladogram from the ancestral cell line, the first living cells, and then the branching into the domain bacteria, domain archaea, and domain eukarya. Interestingly enough, archaea and eukarya actually have more in common than the archaea and the bacteria do with each other. So here's the 3 domains. Archaea, they are prokaryotic cells. Their cell walls contain no peptidoglycan, which is a specific protein found only in bacterial cell walls. Their lipid membranes are composed of branched carbon chains attached to glycerol by ether linkages, which I know doesn't mean anything to you all, but actually is one of the reasons that archaea was separated from bacteria. The ribosomal RNA loop is lacking in the archaea small subunit of RNA and present in the bacterial small subunit of the ribosome in bacteria. The eukarya are all eukaryotic cells. If they have a cell wall, it's containing a carbohydrate, not a protein, such as the peptidoglycan. The membrane lipids are composed of straight carbon chains attached to glycerol, again by ester linkages, and you'll see that this is common throughout all of the domains, is that we have the bilayer of phospholipids in the cell membranes. The eukarya also lack that RNA loop in the ribosome small subunit, and so again, they're more closely related RNA-wise to the archaea than they are the bacteria. So final concept for Chapter 1. How are the names of bacteria assigned and abbreviated? What are the 8 taxonomic categories? What were the 5 kingdoms of the Whittaker system of classification, and into what domain do they now fall? And what are the 3 domains of the Wuest-Fox system that was adopted in 1978 that we currently operate under? And again, taxonomy is constantly changing with our technology. And so this is the last of Chapter 1. I will state here that Chapter 2 is a review chapter over the atom, the cell structures, and some of the other concepts that you learn in anatomy and physiology, and therefore I'm not going to do an actual narrated PowerPoint of Chapter 2. You can simply look over that in your PowerPoints that are in Canvas. They will be testable material. However, I'm not going to lecture if we get to lecture in person over Chapter 2. So be aware of that. Thank you. Have a wonderful day.